A gene is a basic physical and functional unit of heredity. The concept of gene was first explained by Graeber Mendel in 1860s. He used the term gene. He was called it as factor in 1909. The Danish biologist Wilhelm Johannsen coined the term gene that was referred to discrete determinants of inherited characteristics. According to the classical concept of gene introduced by Sutton in 1902, genes have been defined as discrete um, particles that follow Mendelian rules of inheritance, occupy a definite locus in the chromosome, and are responsible for the expression of a specific phenotype character. They show the following properties. Number of genes in each organism is more than the number of chromosomes, and several genes are located on the same chromosome. The genes are arranged in a single linear order like beads on a string. Each gene occupies a specific position called locus. Genes may exist in several alternate forms called alleles. Genes may undergo sudden change in positions and compositions called mutations. Genes are capable of self-duplication, producing their own copies. As early as the experiments in early as 1848, Wilhelm Hofmeister, a German botanist, had observed that cell nuclei organize themselves into small rod-like bodies called during mitosis called chromosomes. In 1869, Friedrich Miescher, a Swiss physician, isolated a substance from cell nuclei and called it as nuclein. It was renamed as nucleic acid by Altman in 1889 and is now known as DNA. By 1920, it became clear that the chromosomes are made up of proteins and DNA. Many experiments were carried out to study the actual carriers of genetic information. Griffith's experiment proved that DNA is the genetic material which has been dealt in class 11. Bacterial transformation experiments provided the first proof that DNA is the genetic material. However, he could not understand the cause of bacterial transformation, and the biochemical nature of genetic material was not derived from his experiments. Later, Oswald Avery, Colin McLeod, and McLean McCarthy in 1944 repeated Griffith's experiments in an in vitro system in order to identify the nature of the transforming substance responsible for converting a non-viral stain into viral stain. They observed that the DNA, RNA, and proteins isolated from the yeast killed S strain when added to R strain changed the surface character from rough to smooth and also made them pathogenic. But when the extract was treated with DNase, an enzyme which destroys a DNA, the transforming ability was lost. RNA, an enzyme, RNase, an enzyme that destroys RNA, and proteases, an enzyme which destroys protein, did not affect the transformation. Digestion with DNase inhibited transformation, suggesting that DNA caused the transformation. These experiments suggested that DNA are not proteins, is the genetic materials. The phenomenon by which DNA is isolated from one type of cell A strain when introduced into another type R strain is able to retain some of the properties of the S strain is referred to as the transformation.